Hello. In this video we will be talking about blood, and we'll be looking at the other formed elements of blood that we have not yet talked about. Specifically, we will be talking about the leukocytes and platelets. So leukocytes are also known as white blood cells. They are different from platelets and red blood cells in that they are actually nucleated, meaning that they are true cells. Although they are true cells, they only make up a small fraction of the total blood volume. One characteristic function of leukocytes is that they exhibit positive chemotaxis. This is where they will move towards higher concentration of chemicals. So they have receptors on their membranes for these different chemicals that they're attracted to, and they'll start moving towards them. The phenomenon of leukocytosis is when you have larger than normal amounts of white blood cells. So the white blood cell counts of over 11,000 cells per microliter will be classified as this leukocytosis. Overall, there are two main categories of white blood cells. There are the granulocytes that contain granules and the agranulocytes that lack them. All right, a characteristic of leukocytes is that they have a specific movement. So they go through what is known as immigration or diapedesis. This is the process by which leukocytes exit the blood vessels. Immigration is more of the name for the overall process, while diapedesis is the process where they are actually leaving their vessels. So what happens is that in capillaries and small venules, the leukocytes that you see here, they start to squeeze between the adjacent endothelial cells. So this endothelial cell that you right here, you see there's gaps there. The leukocytes are able to squeeze in between there, and once they're there, they are going to reach the site of injury, infection, or inflammation. So after leaving the bloodstream, the leukocytes typically will do one of two things. Certain ones will settle in fixed positions, such as lymphatic tissue, bone marrow, spleen, or the thymus, while others will move freely through tissue spaces, similar to amoeba, going towards the site of the infection. All right, so let's talk about the different types of leukocytes, starting with the granulocytes. So the granulocytes are relatively large. They typically are going to be short-lived compared to red blood cells. Remember, red blood cells were typically alive for about 120 days or so, or utilized for 120 days. The granulocytes are going to be shorter, more on the time scale of days or maybe weeks. The characteristics of these, so they have granules that stain with either a red acidic dye known as eosin or a blue basic dye. And so the type of stain they take up is going to be determined by what the granules contain. Another characteristic of them are their nuclei, which will typically have lobes and be characteristically shaped. So let's start with the basophils. The basophils, the term means base-loving. That means that they're going to take up the basic dyes, which are going to stain blue. This results in overall having a purplish or blue appearance because the granules take up that blue stain. The nucleus is also going to stain blue in all of these, and you'll see that the basophils have a deep purple nucleus that is typically either S or U-shaped and only really has 1 to 2 constrictions. So as far as the nuclei go, this one's going to be more condensed into just a single area than we'll see with the other granulocytes. The large dark staining granules contain histamine. The function of histamine is to act as an inflammatory chemical. So it leads to an inflammatory response, including acting as a vasodilator to help blood get to the site of infection, and acts as a chemoattractant, typically attracting the other immune cells to it. One thing to note is that basophils are similar to another type of cell known as mast cells, but these things are not equivalent. So mast cells are going to arise from a different cell line entirely, and they have nuclei that are more oval and less lobed than the basophils. How they are similar is that they both have granules that are rich in histamine. So that's one thing. They both are going to bind to IgE antibodies. So the IgE is one of the stimulating factors that lead to release of the histamine. So the histamine and the IgE are things that they share with mast cells. All right, the next type of cell is the eosinophil. These make up only about 2 to 4% of all leukocytes. They're roughly the same size as neutrophils. They are called eosinophils because they are eosin loving. So eosin is a name that we give to an acidic dye that binds to certain things like what these granules are. The granules are going to be, or the nucleus is typically going to show 2 lobes connected by a narrow band, so it's kind of like a horseshoe shape. And then the granules are going to contain digestive enzymes. So they are somewhat like lysosome. So these digestive enzymes help to counteract certain things, especially parasitic worms, such as flatworms and roundworms. The eosinophils reside in loose connective tissues at the same sites that the worms invade, and when encountering a parasite, the eosinophils will typically gather around it, release toxic enzymes from the granules, and the granules then help digest the parasite away. Eosinophils have also been implicated in allergies and asthma. I just wrote over where I'm going to put some additional text. So, and they will also contribute to tissue damage that is characteristic of immune responses. All right. Next up, we have our neutrophils. So neutrophils are the most abundant white blood cells. They're neutral loving because they have the ability to bind to multiple or to both acidic and basic dyes. So they're known as, so they're going to end up with a lilac color because of the mixture of those. You'll notice the granules here are very fine, so they're difficult to see individually. The nuclei is going to contain multiple lobes, so typically 3 to 6 lobes separated by small bridges that connect to them. Because they are multi-lobed, they're known as polymorphonuclear leukocytes. So the big thing to keep in mind about the neutrophils are that they are antimicrobial, meaning that they are going to attack primarily bacteria. Really, I should have said antibacterial, not microbial. So they primarily attack the bacteria, and so when you have an acute bacterial infection like meningitis or appendicitis, they are typically going to be elevated and can make up a higher percentage of your leukocytes in those instances. They also can act as phagocytes for bacteria and fungi. Right, this brings us to the agranulocytes. These are derived from a slightly different cell line than most of the granulocytes, although there might be a common ancestor between one of them. What characterizes these things from the others is that they lack the cytoplasmic granules. So first of all, we have the lymphocytes. So these can account for about 25% of the white blood cells. Most of these are going to reside in lymphoid tissues. So second most numerous leukocyte. The nucleus takes up most of the volume of the cell, and they can come in different sizes. Ultimately, they're going to play a crucial role in immunity, especially playing an important role in the adaptive immunity. Two major types, so we have T cells. The T cells act directly against virus-infected cells and tumor cells, and they're a key player in cell-mediated immunity. The other type are B cells or B lymphocytes. These are the ones that are going to be responsible for producing antibodies. And we'll talk more about these when we talk about the immune system. Monocytes account for 3 to 8% of the white blood cells. They are the largest white blood cells, so they appear pale blue in the cytoplasm. You'll notice that they have a kidney-shaped nucleus. They are cells that will transform into what are known as macrophages, or big eaters. So when they are in the blood, they are monocytes, but when they move into tissues, they become these mobile phagocytic cells that defend against viruses, intracellular bacteria, and chronic infections. They also are going to help activate lymphocytes. So they are going to help link what is known as the innate immune system to the adaptive immune system. White blood cells are produced through a process known as leukopoiesis. So this is kind of like erythropoiesis, except we're talking about white blood cells. It is stimulated by chemical messengers in the form of paracrine factors or hormones. The paracrines and the hormones are mostly glycoproteins that are going to belong to one of two families. So we have the interleukins these are numbered, typically IL-3, IL-5, etc. And the other family are the colony stimulating factors. These are typically named after the cells they stimulate. For example, GCSF is going to stimulate granulocyte production. These paracrine and hormonal factors, they are going to be produced by supporting cells in the red bone marrow and by white blood cell precursors. Or sorry, they stimulate white blood cells. They are secreted by mature, so they stimulate white blood cell precursors. So the function of these things are to stimulate division and maturation of the white blood cell precursors. They also make the mature leukocytes more effective. So the overall process of it, so we start off with the hematopoietic stem cell that is ultimately going to give rise to a cell that can differentiate into a myeloid stem cell or a lymphoid stem cell. The myeloid stem cell is going to be responsible for giving you your platelets down here. We'll talk about that here in a moment. The red blood cells that we've talked about in previous videos, and then likely two of the granulocytes. So you can see that we have the basophil and the eosinophil here, while the neutrophil, once thought to be sharing a common precursor with these cells, is now believed to actually share a precursor with the monocyte, which is going to be derived from the lymphoid stem cell. So it actually appears that this is the more likely pathway and not that separates those. The monocytes, which form the macrophages, and then the lymphocytes, which these are the natural killer cells. We'll talk about those when we talk about the immune system. But the lymphocyte are going to give rise to either T cells or B cells. And then B cells can form memory B cells, and T cells can also do that. But then these may also form plasma cells that secrete antibodies. All right, so that is the lineage of the white blood cells. Let's go back now and talk about platelets. So platelets are not true cells. They are derived from megakaryocytes. The megakaryocytes can be up to 60 micrometers in diameter, and they fragment off, giving you the platelets that are much smaller, about 1/4 the size of the lymphocytes. In blood smears, they appear to have a blue staining outer region and a purple staining inner region. This purple staining inner region is going to contain granules, Those granules contain things like serotonin, which I'll abbreviate as 5-HT, which is another name for it, calcium, various enzymes, also will contain adenosine diphosphate, and one other thing is platelet-derived growth factor. So these are all going to play a role in the function of platelets, which play an important role in blood clotting, which we'll talk about in another video. The platelet lifespan is relatively short, so if not used in clotting, they last only about 10 days. They are going to be kept inactive by nitric oxide, or NO, and prostacyclin, things that are produced by the endothelial cells that you see here. So those are producing those things and preventing the platelets from clotting. As far as regulation is concerned, they are regulated by thrombopoietin. So thrombopoietin regulates their formation. We've already talked about how the megakaryocytes are formed. So these are going to come from the myeloid lineage. Megakaryocytopoiesis, that's a big name, but ultimately it is the formation of platelets. The process is going to occur within the bone marrow, where the megakaryocytes will undergo repeated cytokinesis, but without mitosis. So you have portions of the regions forming out. So the platelets are going to form as the megakaryocytes press against the sinusoid, which is a special capillary in the red marrow. It'll send these cytoplasmic extensions through the sinusoid wall. And the extensions break apart and form platelet fragments. Although this is the typical way, not all of them are going to be produced this way. Some platelets are produced in the lungs, and this is due to the turbulent blood flow that's shredding whole megakaryocytes into fragments. And it's thought that maybe up to half, or maybe even slightly over half, of the circulating platelets may actually be produced this way. All right, so that was platelets. The last video we'll have in blood will talk about the process of blood hemostasis, blood clotting, etc.